In Arabidopsis thaliana, a common angiosperm, a group of scientists from the University of Lausanne in Switzerland in 2011 wanted to find out what proteins were involved in the Casparian strip formation and whether these formations were related to the construction of the strip. Even though there has been a lot of research in the properties of these cells in the Casparian strip, the mechanisms that cause the cells to form are largely unknown. The Casparian strip is important because it forms belts of cell wall-like material in the root endodermis of plants that assist the plant in the uptake of nutrients, water, and filter toxic substances. In these cell walls, the strips are composed of polymers that prevent the diffusion of water and solids between cells. To do this, Rapallo and his research team first assumed that messenger RNAs, the copy of the DNA in the plant that encode the proteins in the Casparian strip, were found in the endodermis. They used the model plant Arabidopsis thaliana and assessed the mRNA transcript in the root endodermis and identified 5 proteins in a previously undiscovered protein family. They did this by searching for microarrays, which is a collection of microscopic DNA in spots attached to a solid surface, and used this to work out the gene expression amount. They then used this to find out if the proteins were linked to the formation of the strip. Rapallo's team fused the proteins to green fluorescent protein, which literally lights up green under the light, to determine the location where the proteins are expressed in the root endodermis. They found that 5 proteins gathered exactly at the Casparian strip and named these strip proteins Cas1 to 5. However, their research was mainly based on Cas1. To test if the Cas proteins were responsible for the formation of the membrane, the researchers used live imaging of the protein at different stages of formation to see if they formed a strip at 3-minute frame rate and a total duration of 120 minutes. At first, the protein showed uniform distribution in the plasma membrane with a weak green fluorescent protein signal. As time progresses, gradual relocation and patching at the future site of the Casparian strip formation indicated that the Cas are not locating to the previously established domain but are suggesting the formation. The fluorescent protein light increased and displayed the strip formation. They performed further experiments to try and better understand the 5 identified Cas, primarily through mutant knockouts, but their exact roles and mechanisms remain unclear and further experimentation is required. The research project showed that there is a clear role for the proteins in the Casparian strip to construct diffusion barriers, but there are still no defined factors that control their positions.